Medical genetics. The study of the etiology, pathogenesis, and natural history of diseases and disorders that are at least partially genetic in origin. Or, medical genetics is the branch of medicine that involves the diagnosis and management of hereditary disorders. Medical genetics is the branch of medicine that involves the diagnosis and management of hereditary disorders. Medical genetics differs from human genetics in that human genetics is a field of scientific research that may or may not apply to medicine, while medical genetics refers to the application of genetics to medical care. For example, research on the causes and inheritance of genetic disorders would be considered within both human genetics and medical genetics, while the diagnosis, management, and counseling people with genetic disorders would be considered part of medical genetics. In contrast, the study of typically non-medical phenotypes such as the genetics of eye color would be considered part of human genetics, but not necessarily relevant to medical genetics, except in situations such as albinism. Genetic medicine is a newer term for medical genetics and incorporates areas such as gene therapy, personalized medicine, and the rapidly emerging new medical specialty, predictive medicine. Contents. 1. Scope. 1.1 Subspecialties. 1.1.1 Clinical genetics. 1.1.2 Metabolic/biochemical genetics. 1.1.3 Cytogenetics. 1.1.4 Molecular genetics. 1.1.5 Mitochondrial genetics. 2. Genetic counseling. 3. History. 4. Current practice. 4.1 Diagnostic evaluation. 4.1.1 Chromosome studies. 4.1.2 Basic metabolic studies. 4.1.3 Molecular studies. 4.2 Treatments. 4.2.1 Management of metabolic disorders. 4.2.2 Other examples. Scope. Medical genetics encompasses many different areas, including clinical practice of physicians, genetic counselors, and nutritionists, clinical diagnostic laboratory activities, and research into the causes and inheritance of genetic disorders. Examples of conditions that fall within the scope of medical genetics include birth defects and dysmorphology, intellectual disabilities, autism, mitochondrial disorders, skeletal dysplasia, connective tissue disorders, cancer genetics, and prenatal diagnosis. Medical genetics is increasingly becoming relevant to many common diseases. Overlaps with other medical specialties are beginning to emerge, as recent advances in genetics are revealing etiologies for morphologic, endocrine, cardiovascular, pulmonary, ophthalmologist, renal, psychiatric, and dermatologic conditions. The medical genetics community is increasingly involved with individuals who have undertaken elective genetic and genomic testing. Subspecialties. In some ways, many of the individual fields within medical genetics are hybrids between clinical care and research. This is due in part to recent advances in science and technology, for example, see the Human Genome Project, that have enabled an unprecedented understanding of genetic disorders. Clinical genetics. Clinical genetics is the practice of clinical medicine with particular attention to hereditary disorders. Referrals are made to genetics clinics for a variety of reasons, including birth defects, developmental delay, autism, epilepsy, short stature, and many others. Examples of genetic syndromes that are commonly seen in the genetics clinic include chromosomal rearrangements, Down syndrome, DiGeorge syndrome, 22q11.2 deletion syndrome, Fragile X syndrome, Marfan syndrome, neurofibromatosis, Turner syndrome, and Williams syndrome. In the United States, doctors who practice clinical genetics are accredited by the American Board of Medical Genetics and Genomics, ABMGG, that, 1. In order to become a board-certified practitioner of clinical genetics, a physician must complete a minimum of 24 months of training in a program accredited by the ABMGG. Individuals seeking acceptance into clinical genetics training programs must hold an MD or DO degree, or their equivalent, and have completed a minimum of 24 months of training in an ACGME-accredited residency program in internal medicine, pediatrics, obstetrics and gynecology, or other medical specialty. 2. Metabolic/biochemical genetics.
Metabolic or biochemical genetics involves the diagnosis and management of inborn errors of metabolism in which patients have enzymatic deficiencies that perturb biochemical pathways involved in metabolism of carbohydrates, amino acids, and lipids.
Examples of metabolic disorders include galactosemia, glycogen storage disease, lysosomal storage disorders, metabolic acidosis, peroxisomal disorders, phenylketonuria, and urea cycle disorders. Cytogenetics. Cytogenetics is the study of chromosomes and chromosome abnormalities. While cytogenetics historically relied on microscopy to analyze chromosomes, new molecular technologies such as array comparative genomic hybridization are now becoming widely used. Examples of chromosome abnormalities include aneuploidy, chromosomal rearrangements, and genomic deletion/duplication disorders. Molecular genetics. Molecular genetics involves the discovery of and laboratory testing for DNA mutations that underlie many single-gene disorders. Examples of single-gene disorders include achondroplasia, cystic fibrosis, Duchenne muscular dystrophy, hereditary breast cancer, BRCA1/2, Huntington disease, Marfan syndrome, Noonan syndrome, and Rett syndrome. Molecular tests are also used in the diagnosis of syndromes involving epigenetic abnormalities, such as Angelman syndrome, Beckwith-Weidman syndrome, Prader-Willi syndrome, and uniparental disomy. Mitochondrial genetics. Mitochondrial genetics concerns the diagnosis and management of mitochondrial disorders, which have a molecular basis but often result in biochemical abnormalities due to deficient energy production. There exists some overlap between medical genetic diagnostic laboratories and molecular pathology. Genetic counseling. Genetic counseling is the process of providing information about genetic conditions, diagnostic testing, and risks in other family members, within the framework of non-directive counseling. Genetic counselors are non-physician members of the medical genetics team who specialize in family risk assessment and counseling of patients regarding genetic disorders. The precise role of the genetic counselor varies somewhat depending on the disorder. When working alongside geneticists, genetic counselors normally specialize in pediatric genetics, which focuses on developmental abnormalities present in newborns, infants, or children. The major goal of pediatric counseling is attempting to explain the genetic basis behind the child's developmental concerns in a compassionate and articulated manner that allows the potentially distressed or frustrated parents to easily understand the information. As well, genetic counselors normally take a family pedigree, which summarizes the medical history of the patient's family. This then aids the clinical geneticist in the differential diagnosis process and help to determine which further steps should be taken to help the patient. 3. History. Although genetics has its roots back in the 19th century with the work of the Bohemian monk Gregor Mendel and other pioneering scientists, human genetics emerged later. It started to develop, albeit slowly, during the first half of the 20th century. Mendelian, single-gene, inheritance was studied in a number of important disorders such as albinism, brachydactyly, short fingers and toes, and hemophilia. Mathematical approaches were also devised and applied to human genetics. Population genetics was created. Medical genetics was a late developer, emerging largely after the close of World War II, 1945, when the eugenics movement had fallen into disrepute. 4. The Nazi misuse of eugenics sounded its death knell. 5. Shorn of eugenics, a scientific approach could be used and was applied to human and medical genetics. Medical genetics saw an increasingly rapid rise in the second half of the 20th century and continues in the 21st century. Current practice. The clinical setting in which patients are evaluated determines the scope of practice, diagnostic, and therapeutic interventions. For the purposes of general discussion, the typical encounters between patients and genetic practitioners may involve referral to an outpatient genetics clinic, pediatric, adult, or combined, or an in-hospital consultation, most often for diagnostic evaluation. Specialty genetics clinics focusing on management of inborn errors of metabolism, skeletal dysplasia, or lysosomal storage diseases. Referral for counseling in a prenatal genetics clinic to discuss risks to the pregnancy, advanced maternal age, teratogen exposure, family history of a genetic disease, test results, abnormal maternal serum screen, abnormal ultrasound, and/or options for prenatal diagnosis, typically non-invasive prenatal screening, diagnostic amniocentesis or chorionic villus sampling. Multidisciplinary specialty clinics that include a clinical geneticist or genetic counselor, cancer genetics, cardiovascular genetics, craniofacial or cleft lip/palate, hearing loss clinics, muscular dystrophy/neurodegenerative disorder clinics.
Diagnostic evaluation.
Each patient will undergo a diagnostic evaluation tailored to their own particular presenting signs and symptoms.
The geneticist will establish a differential diagnosis and recommend appropriate testing. These tests might evaluate for chromosomal disorders, inborn errors of metabolism, or single gene disorders. Chromosome studies.
Chromosome studies are used in the general genetics clinic to determine a cause for developmental delay/mental retardation, birth defects, dysmorphic features, and/or autism. Chromosome analysis is also performed in the prenatal setting to determine whether a fetus is affected with aneuploidy or other chromosome rearrangements. Finally, chromosome abnormalities are often detected in cancer samples. A large number of different methods have been developed for chromosome analysis. Chromosome analysis using a karyotype involves special stains that generate light and dark bands, allowing identification of each chromosome under a microscope. Fluorescence in situ hybridization, FISH, involves fluorescent labeling of probes that bind to specific DNA sequences, used for identifying aneuploidy, genomic deletions or duplications, characterizing chromosomal translocations and determining the origin of ring chromosomes. Chromosome painting is a technique that uses fluorescent probes specific for each chromosome to differentially label each chromosome. This technique is more often used in cancer cytogenetics, where complex chromosome rearrangements can occur. Array comparative genomic hybridization is a newer molecular technique that involves hybridization of an individual DNA sample to a glass slide or microarray chip containing molecular probes, ranging from large 200 kb bacterial artificial chromosomes to small oligonucleotides, that represent unique regions of the genome. This method is particularly sensitive for detection of genomic gains or losses across the genome but does not detect balanced translocations or distinguish the location of duplicated genetic material, for example, a tandem duplication versus an insertional duplication. Basic metabolic studies. Biochemical studies are performed to screen for imbalances of metabolites in the bodily fluid, usually the blood, plasma/serum, or urine, but also in cerebrospinal fluid, CSF. Specific tests of enzyme function, either in leukocytes, skin fibroblasts, liver, or muscle, are also employed under certain circumstances. In the U.S., the newborn screen incorporates biochemical tests to screen for treatable conditions such as galactosemia and phenylketonuria, PKU. Patients suspected to have a metabolic condition might undergo the following tests. Quantitative amino acid analysis is typically performed using the ninhydrin reaction, followed by liquid chromatography to measure the amount of amino acid in the sample, either urine, plasma/serum, or CSF. Measurement of amino acids in plasma or serum is used in the evaluation of disorders of amino acid metabolism such as urea cycle disorders, maple syrup urine disease, and PKU. Measurement of amino acids in urine can be useful in the diagnosis of cystinuria or renal Fanconi syndrome, as can be seen in cystinosis. Urine organic acid analysis can be either performed using quantitative or qualitative methods, but in either case the test is used to detect the excretion of abnormal organic acids. These compounds are normally produced during bodily metabolism of amino acids and odd-chain fatty acids, but accumulate in patients with certain metabolic conditions. The acyl-carnitine combination profile detects compounds such as organic acids and fatty acids conjugated to carnitine. The test is used for detection of disorders involving fatty acid metabolism, including CAD. Pyruvate and lactate are byproducts of normal metabolism, particularly during anaerobic metabolism. These compounds normally accumulate during exercise or ischemia, but are also elevated in patients with disorders of pyruvate metabolism or mitochondrial disorders. Ammonia is an end product of amino acid metabolism and is converted in the liver to urea through a series of enzymatic reactions termed the urea cycle. Elevated ammonia can therefore be detected in patients with urea cycle disorders, as well as other conditions involving liver failure. Enzyme testing is performed for a wide range of metabolic disorders to confirm a diagnosis suspected based on screening tests. Molecular studies. DNA sequencing is used to directly analyze the genomic DNA sequence of a particular gene. In general, only the parts of the gene that code for the expressed protein, exons, and small amounts of the flanking untranslated regions and introns are analyzed. Therefore, although these tests are highly specific and sensitive, they do not routinely identify all of the mutations that could cause disease. DNA methylation analysis is used to diagnose certain genetic disorders that are caused by disruptions of epigenetic mechanisms such as genomic imprinting and uniparental disomy. Southern blotting is an early technique basic on detection of fragments of DNA separated by size through gel electrophoresis and detected using radiolabeled probes. This test was routinely used to detect deletions or duplications in conditions such as Duchenne muscular dystrophy but is being replaced by high-resolution array comparative genomic hybridization techniques. Southern blotting is still useful in the diagnosis of disorders caused by trinucleotide repeats. Treatments. Each cell of the body contains the hereditary information, DNA, wrapped up in structures called chromosomes. Since genetic syndromes are typically the result of alterations of the chromosomes or genes, there is no treatment currently available that can correct the genetic alterations in every cell of the body. Therefore, there is currently no cure for genetic disorders. However, for many genetic syndromes there is treatment available to manage the symptoms. In some cases, particularly inborn errors of metabolism, the mechanism of disease is well understood and offers the potential for dietary and medical management to prevent or reduce the long-term complications. In other cases, infusion therapy is used to replace the missing enzyme. Current research is actively seeking to use gene therapy or other new medications to treat specific genetic disorders. Management of metabolic disorders. In general, metabolic disorders arise from enzyme deficiencies that disrupt normal metabolic pathways. For instance, in the hypothetical example, A B C D quad A B B B B B B C C C C C C C C C C no D X Y Z X Y no or insufficient Z E. Compound A is metabolized to B by enzyme X, compound B is metabolized to C by enzyme Y, and compound C is metabolized to D by enzyme Z. If enzyme Z is missing, compound D will be missing, while compounds A, B, and C will build up. The pathogenesis of this particular condition could result from lack of compound D, if it is critical for some cellular function, or from toxicity due to excess A, B, and or C, or from toxicity due to the excess of E which is normally only present in small amounts and only accumulates when C is in excess. Treatment of the metabolic disorder could be achieved through dietary supplementation of compound D and dietary restriction of compounds A, B, and or C, or by treatment with a medication that promoted disposal of excess A, B, C, or E. Another approach that can be taken is enzyme replacement therapy, in which a patient is given an infusion of the missing enzyme Z or cofactor therapy to increase the efficacy of any residual Z activity. Diet. Dietary restriction and supplementation are key measures taken in several well-known metabolic disorders, including galactosemia, phenylketonuria, PKU, maple syrup urine disease, organic acidureas, and urea cycle disorders. Such restrictive diets can be difficult for the patient and family to maintain, and require close consultation with a nutritionist who has special experience in metabolic disorders. The composition of the diet will change depending on the caloric needs of the growing child, and special attention is needed during a pregnancy if a woman is affected with one of these disorders. Medication. Medical approaches include enhancement of residual enzyme activity, in cases where the enzyme is made but is not functioning properly, inhibition of other enzymes in the biochemical pathway to prevent buildup of a toxic compound, or diversion of a toxic compound to another form that can be excreted. Examples include the use of high doses of pyridoxine, vitamin B6, in some patients with homocystinuria to boost the activity of the residual cystathione synthase enzyme, administration of biotin to restore activity of several enzymes affected by deficiency of biotinidase, treatment with NTBC and tyrosinemia to inhibit the production of succinylacetone which causes liver toxicity, and the use of sodium benzoate to decrease ammonia buildup in urea cycle disorders. Enzyme replacement therapy. Certain lysosomal storage diseases are treated with infusions of a recombinant enzyme, produced in a laboratory, which can reduce the accumulation of the compounds in various tissues. Examples include Gaucher disease, Fabry disease, mucopolysaccharidosis, and glycogen storage disease type 2. Such treatments are limited by the ability of the enzyme to reach the affected areas. The blood-brain barrier prevents enzyme from reaching the brain, for example, and can sometimes be associated with allergic reactions. The long-term clinical effectiveness of enzyme replacement therapies vary widely among different disorders. Other examples: angiotensin receptor blockers in Marfan syndrome and Loeys-Dietz, bone marrow transplantation, gene therapy. Medical genetics is any application of genetic principles to medical practice. This includes studies of inheritance, mapping disease genes, diagnosis and treatment, and genetic counseling. Pharmacogenetics is the study of how drugs affect the body with respect to specific genetic backgrounds. Knowledge of these effects can improve effectiveness of drugs and minimize side effects on an individual patient basis. Genetic diseases, disorder definition, types, and examples. Definition. Types. Single inheritance. Multifactorial inheritance. Chromosome abnormalities. Mitochondrial inheritance. What is a genetic disease?
Abnormalities in an individual's genetic makeup cause genetic disease.
Abnormalities in an individual's genetic makeup cause genetic disease. A genetic disease is any disease caused by an abnormality in the genetic makeup of an individual. The genetic abnormality can range from minuscule to major, from a discrete mutation in a single base in the DNA of a single gene to a gross chromosomal abnormality involving the addition or subtraction of an entire chromosome or set of chromosomes. Some people inherit genetic disorders from the parents, while acquired changes or mutations in a pre-existing gene or group of genes cause other genetic diseases. Genetic mutations can occur either randomly or due to some environmental exposure. What are the 4 types of genetic disorders, inherited? There are a number of different types of genetic disorders, inherited, and include: 1. Single gene inheritance. 2. Multifactorial inheritance. 3. Chromosome abnormalities. 4. Mitochondrial inheritance. 6. Symptoms and signs of Down syndrome. The baby with Down syndrome has a hallmark appearance. However, every aspect of the appearance does not need to be present, as the phenotype, the way the genes make the child look, can be markedly different for each patient. Common Down syndrome symptoms are: 1. A small head and short neck. 2. Flat face, and 3. Upward slanting eyes, ears are flat and position lower than normal. 4. The tongue protrudes and seems to be too large for the mouth. 5. Hands tend to be wide. 6. Short fingers and there is just a single flexion crease in the palm, and 7. Joints tend to be more flexible and muscles may lack tone. 7. Single gene inheritance disorders. Single gene inheritance is also called Mendelian or monogenetic inheritance. Changes or mutations that occur in the DNA sequence of a single gene cause this type of inheritance. There are thousands of known single gene disorders. These disorders are known as monogenetic disorders, disorders of a single gene. Single gene disorders have different patterns of genetic inheritance, including autosomal dominant inheritance, in which only one copy of a defective gene, from either parent, is necessary to cause the condition. Autosomal recessive inheritance, in which 2 copies of a defective gene, one from each parent, are necessary to cause the condition, and X-linked inheritance, in which the defective gene is present on the female or X chromosome. X-linked inheritance may be dominant or recessive. Some examples of single-gene disorders include 1. Cystic fibrosis. 2. Alpha and beta thalassemias. 3. Sickle cell anemia, sickle cell disease. 4. Marfan syndrome. 5. Fragile X syndrome. 6. Huntington's disease, and 7. Hemochromatosis. 7 common multifactorial genetic inheritance disorders. Readers' comments 8. Share your story. Multifactorial inheritance is also called complex or polygenic inheritance. Multifactorial inheritance disorders are caused by a combination of environmental factors and mutations in multiple genes. For example, different genes that influence breast cancer susceptibility have been found on chromosomes 6, 11, 13, 14, 15, 17, and 22. Some common chronic diseases are multifactorial disorders. Examples of multifactorial inheritance include 1. Heart disease. 2. High blood pressure. 3. Alzheimer's disease. 4. Arthritis. 5. Diabetes. 6. Cancer, and 7. Obesity. Multifactorial inheritance also is associated with heritable traits such as fingerprint patterns, height, eye color, and skin color. For chromosomal abnormalities. Chromosomes, distinct structures made up of DNA and protein, are located in the nucleus of each cell. Because chromosomes are the carriers of the genetic material, abnormalities in chromosome number or structure can result in disease. Chromosomal abnormalities typically occur due to a problem with cell division. For example, Down syndrome, sometimes referred to as Down syndrome, or trisomy 21 is a common genetic disorder that occurs when a person has 3 copies of chromosome 21. There are many other chromosomal abnormalities including 1. Turner syndrome, 45AX0. 2. Klinefelter syndrome, 47XXY, and 3. Cri-du-Chat syndrome, or the cry of the cat syndrome, 46XX or XY, 5P. Diseases may also occur because of chromosomal translocation in which portions of 2 chromosomes are exchanged. 3. Mitochondrial genetic inheritance disorders. This type of genetic disorder is caused by mutations in the non-nuclear DNA of mitochondria. Mitochondria are small round or rod-like organelles that are involved in cellular respiration and found in the cytoplasm of plant and animal cells. Each mitochondrion may contain 5 to 10 circular pieces of DNA. Since egg cells, but not sperm cells, keep their mitochondria during fertilization, mitochondrial DNA is always inherited from the female parent. Examples of mitochondrial disease include 1. Leber's hereditary optic atrophy, LHON, and eye disease. 2. Myoclonic epilepsy with ragged red fibers, MRRF, and 3. Mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes, MELAS, a rare form of dementia.